Hello.
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 Hi, how are you? Welcome.
Good. I'm good. Thank you.
Thank you so much. We're really excited you're here.
Oh no, pleasure. Pleasure.
So we'll just give it a minute or two as people start to file in.
Sean,
Hello. Hey.
welcome.
Yeah, thank you. Thank you. Nice to see you.
Thank you for coming.
Pleasure.
How is the weather over there? I was seeing a forecast for lots of snow.
It's been pretty cold.
Yes.
It is. We just had in my daughter's school, there was a kind of a school walk to school that we met at the library, and all of the kids did a kind of a walk to school, and it's despite the freezing weather, um, yeah, it was cold. I guess you
Well,
guys get it equally badly up in
we
Michigan.
get it cold, but
Probably
then it,
worse.
it fluctuates between very cold and then 37 degrees and everything melts and then it gets cold again.
Okay, okay,
So
okay.
it can't decide.
No.
All I can say for those of you who were not in Ann Arbor 30 years ago is that it used to be a lot worse,
It did used to be a lot colder for
unfortunately,
sure.
and more snow.
Huh.
In the 90s, it was not uncommon to have 2 feet, 3 feet, you know.
Yeah, and even into April too. So that has definitely changed. Few more people coming on and then we'll get started here. So we'll go ahead and get started. Hi, everybody. Welcome to our sleep medicine grand rounds today. It's my pleasure to introduce Dr. Sean Purcell, who's an associate professor of psychiatry at Brigham and Women's Hospital at Harvard University. His first degrees were in experimental psychology from the University of Oxford and University of London, and his PhD is from the Institute of Psychiatry at King's College in London. After completing a fellowship at the Whitehead Institute, he joined the Psychiatric and Neurodevelopmental Genetics Unit at Massachusetts General Hospital in Boston, where his primary focus was in neuropsychiatric genetics, working with colleagues at the Stanley Center for Psychiatric Research and the Broad Institute of Harvard and MIT. At that point, his main project centered around developing statistical and computational tools for the design of genetic studies and the detection of gene variants influencing complex human traits and the dissection of these effects in the larger context of genetic and other environmental factors. He has completed and participated in several landmark genome-wide association and exome sequencing studies of neuropsychiatric disease, particularly bipolar disorder and schizophrenia, and currently studies the links between sleep, behavior, cognition, and psychiatric disease using human genetic and computational approaches with the aim of providing opportunities for precision medicine and targeted interventions in neuropsychiatric disease. So welcome. We are thrilled to have you here and excited to learn from you.
Excellent. Thank you. Thank you. I'm actually going to turn my camera off when I present because my connection's a little flaky here. I apologize in advance.
Sure.
So let me get set up. Share.
Great.
Okay. You
Yeah,
can
we can see that.
Okay. Sorry. Okay, great. Excellent. Thank you. So yeah, as noted, I kind of came to sleep midway my scientific career, but it's been a great home, and yeah, I'm looking forward to chatting about some of my work here today. So I want to touch on two kind of broad areas, really. First, developing objective sleep-based biomarkers for neuropsychiatric and neurocognitive disease, but with particular application to schizophrenia, cognitive aging, and thinking about medication effects as well. And then secondly, some of my work in developing tools and resources for sleep science, in terms of data, but also code and models, which is largely driven by my involvement in the National Sleep Research Resource, which I co-direct at the Brigham. And I'll actually jump back and forth between these two themes during the talk, hopefully with the goal of understanding or underscoring, sorry, some of the ways in which these parallel efforts can hopefully inform each other. So, I don't think any of you would be on the call if you believe that maintaining healthy sleep plays an important role in maintaining physical and cognitive health more generally. So I don't want to belabor any of these points, but rather just highlight my own areas of interest, which isn't on sleep disorders per se, but primarily on brain activity during sleep and its relation to cognitive and psychiatric outcomes. I view sleep as a powerful and potentially unique window on 24/7 pathology, such that, even if some diseases such as schizophrenia are not fundamentally caused by sleep problems or irregularities per se, I don't think there's a direct link from sleep phenotypes to psychiatric symptoms in this case. But still, I'd like to make the case that studying sleep-based biomarkers can nonetheless be informative and offer kind of a potentially unique window. So just stepping back, some just very general kind of points right about measurement. When we talk about sleep, right? What do I mean? So stepping back, obviously we know we can measure sleep in several different ways, right? From subjective report, PSG-derived macro architecture, or from actigraphy, wake sleep cycles, dividing the night into classical stages, and so on. But then look at the EEG as a kind of a more direct readout of neural activity, and as I said, my primary focus. So the PSG is typically taken as the gold standard here, implying self-report or actigraphy are in some senses less accurate. But the point I want to make is they really give you qualitatively different types of information. Self-report obviously is subjective, and as such, it's the only way to quantify experiential aspects of sleep, such as daytime tiredness or lack of restorative sleep. And then considering the objective measures, actigraphy is more scalable and can be deployed in naturalistic settings for days or weeks to objectively measure sleep regularity and circadian rhythms. Of course, it doesn't capture the so-called microarchitecture of sleep. And so these approaches give us really three very different representations of the same night's sleep. So macroarchitecture, the timing and so on, and duration of sleep in terms of stage composition here. But I'm really more focused on this kind of the EEG-derived architecture here. So here showing for a single channel, EEG activity across the night here. And, we know that people differ, obviously, in their average, in their typical sleep marker architecture over and above sleep stage or duration as we get from cycle-based analyses. And some of these individual differences are very stable. Some of them are related to demographic effects. And so, just looking at these representations, clearly in a teenager versus two middle-aged adults, these look like two very different, things going on during the night here, just on a kind of a cursory view. And even these two middle-aged adults, they were selected to have similar N2 and REM durations across the night. Clearly, just by eyeballing here, there's differences here in the sleep microarchitecture as well. So how do we quantify this rather than just staring at pictures? So one classical way is just to average over a given stage, and we can extract the so-called power spectrum. This is for N2 sleep and so on for one individual. And, there's a number of things one can do with this. I actually, with a background in quantitative genetics, find these fascinating kind of biomarkers in that they very clearly show both trait-like and genetic sources of variability, but at the same time, that there's clearly a lot of evidence showing that they're highly state-dependent as well. And so, speaking to the heritability piece, this was just an example in a study we had. We happened to have two MZ twin pairs here, one pair at the top, one at the bottom. And just underscoring the fact that, the sleep EEG is sometimes called a so-called EEG fingerprint. The correspondence here, each member of the twin pair is in either the orange or blue, is really quite striking for a complex quantitative phenotype. Versus in contrast, just to illustrate the variability in these markers, individuals demographically matched but unrelated pairs, we see there's a lot of kind of variability person to person here. That again, presumably at least some portion of this is genetically driven as based on twin studies. And as I said, but we know it's dynamic night to night, clearly, reactive to recent activity, stress, medication, circadian phase, and other effects as well. And even within one night, sleep is a dynamic process. And so if we summarize brain activity in various ways from the EEG, one number doesn't capture necessarily what's happening across the whole night. This is just a slightly different way to represent in two EEG channels here, the activity across the night that's perhaps a little clearer rather than the spectrogram at the top. These are so-called Hjort parameters. It's just a way I have of plotting these, where the color and the height of the band is essentially, well, the height of the band is a measure of the amplitude. And so within a non-REM cycle, you can see these very characteristic kind of build-ups and dissipation of EEG power across the night that, occurs in very kind of characteristic ways. So these are all things that we might want to measure. And, another approach is the most kind of granular level is to look at individual transient events. And so, one focus of my own work has been to look at sleep spindle activity. So I'm sure most of you are, highly familiar with this, right? But just characteristics of N2 sleep, little bursts on the order of a second of 10 to 15 hertz activity generated in the thalamus and propagated by a thalamocortical loop. I find it's interesting in that, historically, PSG included an EEG. It looked at brain activity, primarily, really solely just to define sleep objectively. Is somebody awake or asleep? And then can I measure other aspects of their, respiration and so on? Here, it's almost turning that around, and it's using non-REM sleep as a unique window onto individual differences in brain activity to say something about disease. And so this is why we're focused on these kinds of elements. And I guess the question is, you can imagine there's lots of ways one can query these kinds of data. Does it add anything over and above just a simpler quantification of sleep as being long or short, poor, fragmented, or consolidated? And at least I think in my own work, there's suggestion, and I think from other work that it really does add something on top of this. So as one example, we performed a study in just about 3,500 older individuals from the MESA and Mr. Oz cohorts, where the endpoint here was looking at cognitive performance. These are all older adults. And what we found, this is based on a single night of just home-based PSG, that the so-called macro and micro architectures were actually only very loosely interrelated with each other. So just sleep timing, sleep duration, and so on was really only loosely linked to spectral power during non-REM spindle activity and slow oscillation activity and so on. So it really is kind of capturing something kind of distinct, and was also distinct from one's own self-report of an individual's sleep quality, typical sleep quality. So this is a heat map where these white bands, you probably can't see this, I apologize, but the white is really showing this kind of lack of correlation between what we're capturing by looking at EEG microarchitecture during sleep versus the other, perhaps kind of classical ways of measuring sleep. these are the same data just projected in a different way. If we cluster the different classes of variable, we see this very kind of clear structure here of relative independence between these different types of measures. So in other words, just motivating, the EEG was only here just to stage sleep, but this convinces me that we're not entirely wasting our time by diving into it and seeing what additional information that's relevant to health or disease can we extract from these signals. So with that as a kind of a background, the application here towards schizophrenia. So this was a study that
I'm sorry, can I
Oh yeah,
ask
sure.
I just put it in the chat, but I just wanted to, can we go back to that previous slide with the
Yeah.
architecture?
Sorry, I
So-
didn't. I half saw the chat. So just if people have questions, which is great, please just shout out because I might not see the chat always.
Yeah, so, I mean, even without looking at, spectrum, when we see changes or people will actually clinically ask this
Yep.
question, one about the sleep quality, but the point is like, so we have this, but what can somebody do about it, right? I
Uh-huh.
mean, do we have any other than, treating sleep apnea?
Yep.
So, I mean, has anyone looked at ways to actually sort of
They
try to
have.
change or improve?
Yeah. They have. So I won't speak a lot to that, but we can loop back to it at the end. I'll kind of speak to this a little bit in about a slide or two, a couple of slides, the kind of the so what question, right? So I'll maybe let me get to that, and because it's a great question, obviously, and I think there's a number of answers. Some are direct and some are less direct, but I'll give you my perspective at least on what I'm thinking of doing. So it's a good question, but we just put a pin in that for now, and we'll come back to that.
Okay, thank you.
No, thank you. Okay, so with non-REM markers of schizophrenia. And so this was a project with colleagues at the Broad Institute and the sample collection in China. And really the goal was to try and identify these stable, objective neurophysiological biomarkers, ideally to stratify patients, right, based upon, in this case, thalamocortical function. So we had high-density sleep EEG, wake ERPs, cognitive and clinical measures, as well as some whole genome and exome sequencing. This data freeze had on the order of like 300 individuals in total. We're extending this into bipolar and it's an ongoing study. So we originally published the first view on this back in 2022. Where, the first goal, there had been a prior literature with numerous studies finding a spindle deficit in patients with schizophrenia. And some of these were, there are a number of studies and some really good ones, but a number of these were very small studies. And so we wanted to start off just by, at least in a larger sample, unambiguously demonstrating the presence of a spindle deficit and more broadly then characterizing, what else is varying here in patients versus controls as a first step. And so we were able to do that. I'm not going to step through the results, in any kind of level of detail. I want to kind of focus on a higher level of just, how I think about biomarkers and how they may be used. And so, in this case, it's a spindle deficit and other things to do with their morphology, their coupling of slow oscillations, some aspects of functional connectivity during sleep and so on. But we can think of that as somewhat black box for the purpose of this talk. Okay, these are sleep-based markers that track with case control status. But it was interesting, actually, that the sleep EEG effects were broadly statistically independent of the waking ERP effects. There's a long literature in schizophrenia just looking at these kind of evoked response potentials in the EEG in various paradigms. And so we were kind of curious, are these measuring, some of the same things as sleep is measuring in terms of the spindle deficit? And the answer seemed to be no to a first approximation. As a proof of principle, really, in this paper, and then we subsequently replicated, just recently published in Sleep, in an expanded sample. Can we take these collectively and just at least predict case control status, right, diagnosis in this ascertained case control sample? And the answer was yes, we could build a model pretty successfully and with very high AUC of 0.94 in this first wave. Take that same model. The details of this, it doesn't matter really for this, but this was based on a kind of a data reduction across a large number of measures, but doing it in a way that we thought was statistically robust. And indeed, when we projected this into the second independent sample, we retained this kind of good performance. So it would suggest so. So, that's great, but so what again is the question, right? What do we do with that information? And so I think that there are a number, and this speaks to your question, right? What we found is a statistical mean difference in some set of measures between diagnostic groups. And so, a potential risk or potential diagnostic biomarker. But what do we do with that? Because it isn't clear that there's no world in which this is suddenly going to become a practical, at least based on these data alone, kind of diagnostic biomarker for schizophrenia versus the standard way to diagnose disease, right? That really isn't kind of the goal. So, what is the goal? So one line of work that is my colleagues, this isn't my primary work, but is obviously to use these findings as an entry point into neurobiology. Okay, and the PI of mine on this GRIN study is primarily a mouse geneticist, and so as I'm sure many of you know, what's beautiful about looking at some of these measures in humans is that they're highly conserved, not completely conserved, and I think that exact extent of conservation is a kind of an interesting question, but broadly conserved along with the genes in mice as in humans. And so this obviously reflects one entry point to use spindle activity and other aspects detected here in humans as a kind of a readout of activity in the mice. So for example, we had a study where we knocked out a calcium channel gene that's expressed in the TRN, which is the pacemaker for spindles, and indeed found that in knockout mice, you reduce spindle activity. That then, of course, can become the basis for how can we rescue that with novel therapeutics and so on. And so that is a line of characterizing different gene variants and potential therapeutic avenues, using the sleep EEG in mice, but we're trying to make this as carefully informed by the human work as possible, and thinking about these translational kind of gaps. The other, alternatively, right, can you change these? So definitely, I have colleagues working actively on the potential for neuromodulation, either pharmacologically or say via closed loop stimulation, right, to try and entrain spindles, to entrain slow oscillations and so on. And there's a line of research that certainly you can induce these transients, and then people ask, does that lead to, say, improvements of cognition, of sleep-dependent memory consolidation, and so on? I think that the jury is somewhat out on that. Is my take. It's not my primary area of work, but certainly under some causal models, you could imagine that having an effect. How I see a kind of almost an orthogonal rationale for doing all of this is much more about patient stratification, right? In the context of a disease such as schizophrenia that we know is highly heterogeneous, it's highly polygenic, hundreds of thousands of genes of tiny effect. The kind of the diagnostic, the nosology of disease is based largely on historical accident rather than anything etiologically grounded. So in other words, there's massive sharing between different disorders of genetic influences, say between bipolar disorder, even autism and schizophrenia, and then within a diagnostic group, there's a lot of variability between patients, and presumably in terms of their course, in terms of treatment response, and so presumably etiology. And so I think the idea of this is, would this be one source of a biomarker that could potentially stratify patients with respect to some of these more clinically meaningful kind of outcomes that could be perhaps downstream used to guide treatment and so on. So that is the class of broader term goal of what we do with it. Of course, once we start thinking about not just group level differences, but we're starting to think about individual to individual variability, in the context of disease, it immediately just suggests, it's important to understand just in the typical population, what are the sources of variability? What makes somebody look like this and somebody look like that? So we can think of developmental trajectories, within the night, dynamics of the sleep process, depending, where we look at things, medication effects, other state-dependent effects. And these kinds of things will largely be interrelated. And so what I want to try and get at is, alluding to some of these things that you'll see, I'll segue into talking about the NSRR and population data and large data sets, how there's some interplay between trying to understand something about these markers with respect to these kinds of factors, right? How they just normally vary between and within people, and looping that back to how we interpret a kind of a focused disease ascertained study of, in this case, schizophrenia. So that's kind of a broad goal. And indeed, when we look within the schizophrenia study, right, as well as just looking at mean differences, average differences between groups, what I'm talking about is a sleep biomarker potentially, indexing etiologically distinct types of patient, right? It's reasonable to hypothesize that a signature of that would be that you'd expect to see not just a group difference, but among patients, more variability between patients than you see just between controls to controls, right? Precisely because we've got this mixing of different, effectively almost different disorders that are being labeled as the same thing. And so that heterogeneity, right, is something that we think may be manifest in this increased variability among patients. And so we looked at that, right, as well as just looking again at group differences, just among patients, do we see increases in variance? And indeed, we found for some metrics, we found very significant differences in increased variance among patients here compared to controls. But in this case, there is no mean difference. So on average, these were all inpatient studies, I have to say. But in that context, the patients with schizophrenia, on average, the sleep duration was identical to controls. It's just that when you look at them, there are many more that had very long or very short and so on for sleep duration. So that's an effect that we weren't otherwise capturing at all, right, in just that kind of group level analysis. But clearly something is going on here that we should try and understand. If we look at other metrics, we see qualitatively different patterns. So some metrics where there's a difference in mean, but not in variance, and some metrics where there's a difference here in both. And so, yeah, this is something we'll kind of return to, but understanding what are the sources of variability in both groups is going to be key here. And so this is where we'll return to this, but this is where I kind of bring in some of the kind of population level large data sets that we have in NSRR to try and inform some of this work. So slightly shifting gears, we'll return to the schizophrenia work, as I said. The NSRR is a resource that I founded by Susan Redline in 2014, and I now co-direct it with her. The initial motivation was really data aggregation, as was going to be necessary for large-scale human genetics and epidemiological studies of sleep. But over time, we've really focused on additional kind of trying to add value, essentially, to these data, and to try and encourage the use of data repurposing, to use these for methods development, et cetera, et cetera. And so this URL, sleepdata.org, is where this lives. So yeah, it's been around a while now, and it's still growing. So now there's over 50,000 whole night EDFs on almost 50,000 individuals, most of whom have PSG and EEG. There's some actigraphy, and what's nice about many of these studies is they're large population-based cohort studies, and so there's a large extent of other clinical and demographic information available. In some cases, genetics and omics and other imaging data and so on. This is out of date now. There's certainly over 30 cohorts now, but it's growing. And as I'm sure you know, NIH's somewhat recent data sharing mandate now, if you put in a grant, it has to be, the data have to be shared. NSRR is a kind of an officially blessed repository in that regard. And so we're looking forward to getting much more sleep data in the coming years and trying to expand into both animal and circadian study designs, as well as the kind of traditional focus here on sleep epidemiology studies. Just to give a sense of, I kind of alluded to this, some of the new data that we're kind of getting in This is just a table that, again, it's now probably out of date, but you can see there's a wide range of different kinds of studies here. We're seeing an increase in pediatric data as well, which is nice. So it's quite a diverse data set. And indeed, one I think interesting source of data as well are just archived clinical data that can be, I think, aggregated and really can be quite informative. We've had a few examples of this. There's the Nationwide Children's Hospital has a pediatric data set with full EHR records and so on in the NSRR. We've recently, with colleagues, been involved in collecting some other pediatric clinical studies from a number of sites, and those data will soon be deposited in NSRR, the RASP study. So we have data, that's the core resource, but we try and add value, and this constitutes what I like to call the NSRR's sleep cycle, if you will. So a lot of what I spend time doing is developing tools to try and aid the analysis of many often noisy, often heterogeneous standards and so on across hundreds or thousands of studies. And so that's one place where we try and add value. And then we make an effort to standardize some of the data dictionaries for resources, again, with the goal of making these essentially easier to work with and to perform cross-cohort studies. There's quite an active community outreach component to the NSRR, where we try and put on, say, webinars to talk about new data sets or tools and so on for users. And then ideally, that brings people in and to encourage them to deposit data back into this resource and form this kind of virtuous cycle. As I mentioned, a lot of my work is based on the methods development kind of piece and the tool development piece. And I think there's a real need for this in many ways in sleep science. A colleague of mine, Timur Sofa, put out this little kind of commentary in Sleep last year that was just doing a kind of a review of a given year, looking at all literature published on sleep methods or algorithms in 2019. And the finding here was the vast majority of those papers didn't publish either code, an example, or any data. And when they did, right, it was because the journals were explicitly asking for it. I come from the genetics field originally, where it's much more common just to have open data and open tools and a kind of a robust infrastructure there. And I think it's kind of very key. And so we'd really like to see this improve, because if somebody publishes an interesting method, it's frankly of kind of limited use if other people aren't able to really take and apply and modify and improve that. So to that end, this tool, Luna, I'm not going to go through the details, but we've been developing this and trying to make it better documented, more accessible, say via Python interfaces and so on. But it's effectively designed to work with large numbers of studies from the NSRR and other data sets. And there's a few web apps as well. I can share these links, and these are all available from the websites. But different ways to pull and to view NSRR data and kind of interactively work with it. So what are some of the kind of advantages, if you like, of working with NSRR data? I think some of them are obvious. If it maps onto your research question, right? These data are free, right? Somebody's already collected them. They're relatively large. There's diversity of cohorts. Some of them are longitudinal. There's demographic diversity. Most of these studies aren't ascertained, but there are clinical outcomes, and the PSG is in many cases quite a rich multimodal source of data. As I mentioned, some of these are linked. These, say, the genetic data are housed in other databases, but there are linkages that can be requested with these other sources of information. And I think one of the key applications of NSRR data really has underpinned, I think, probably, if not all, certainly most of the modern advances in automated sleep staging methods that we've seen, in the last kind of couple of years. Clearly, for decades, people have been building automated stagers. I think there's been a little bit of a shift in the last few years, where either because of the methods or just because of the bulk and diversity of the training data, that it's the robustness of these methods has kind of scaled up in a way that is now more usable. We have, at least for research purposes, we have our own variant of this trained on NSRR data that on average performs reasonably well. But then one of the advantages we saw of having a demographically more diverse samples was in some work with a colleague, Natalie Gong, a psychiatry resident at Brigham, finding that in pediatric samples, you do better if you train models on, guess what, on pediatric, manually staged PSGs than if you just use a kind of a cosmopolitan or generic, primarily adult model. So this was just an example of having a better demographically matched training sample, and in this case, getting some additional value from being able to expand to include more channels. But both of those things independently improved the performance here of this stage. And so I think that's kind of one theme to come out of this as well, is the room for potential bias in some of the prediction models, either of staging or of other outcomes, where it's important to think about the population in whom you're applying some method and that things are well aligned. One other just example of the kind of secondary data, kind of low-hanging fruit type of analysis from NSRR data. We had a journal club, an instructor in my group, Dr. Natalia Kashemiakou, and we came across a very nice paper that showed a particular marker, the spectral slope of the EEG in wake and in sleep, and showing that this tracked as a kind of basically a marker of cortical arousal across these sleep states. It was a very nice study. They had imaging data and so on, but the core of the EEG part was on 14 individuals primarily, something like that. So we had our journal club, and just a few days later, really, because the data, was sitting there, we were able to get a preprint out looking in 15,000 studies and finding, replicating in a large sample the same effects, but then being able to leverage the large sample and showing how these things change with age, say, and some additional kind of questions that we were able just to kind of add on by virtue of these larger samples. And so again, with the tools in hand, being able to do this very efficiently, very quickly, I think kind of speaks to the value of working with archival data. Of course, it's not all completely easy going. There are cons, right, with working with convenience data sets. In NSRR, right, perhaps most fundamentally, most of it is human. We're trying to increase the amount of animal data, but that obviously is a limitation. It's mostly scalp EEG, limited PSG montages, and so on from home-based studies. Historically, there was an underrepresentation of healthy younger adults, but that's hopefully changing. But typically, these aren't ascertained on a given disease. And so if you're interested in a rare outcome, this obviously won't be great. Practically, just working with different data sets can be logistically or technically challenging, tedious at best. And so we've tried to, this is partly why we have tools to try and help work with these. But perhaps more importantly, subtle biases can be amplified in large data sets. And, depending upon the original study protocol, those original studies were likely not designed to answer your current research question. And as such, there may be biases that become, very important and relevant. And so it's important to work with these data and understand those properties. Just to try and make that, I know it's a vague statement, to make it somewhat more concrete. This just shows some of the things, if you go looking across, thousands, tens of thousands of hours of unattended sleep-based recordings, of course, you're going to see just a lot of technical artifact and noise. We find all kinds of things of reversed EEG polarities, different hardware filterings impacting things, and so on. So you can kind of somewhat track a lot of this down. But it's important to do QC and look for this stuff. This was one example I thought was interesting just because it impacted perhaps one of the simplest measures to come from studying sleep, total sleep time. Okay, and PSG is the quote-unquote gold standard for doing this. We found that in the sleep heart health study, so there's 5,000 individuals in this first wave, and about half of them make it through to this second wave 5 or 6 years later. And these studies are used in a large number of kind of contexts. And when we actually just looked at what is the total sleep time, we found this very paradoxical kind of inconsistent association with total sleep time and age, where as we'd expect within wave, right, total sleep time declines with increasing age. Yet between wave, right, you still see that same within wave 2, that same decrease, but that between wave, it actually increases within individual. So, that scratched our heads a little bit. That wasn't completely clear. And as far as I'm aware, hasn't been appreciated in people using some of these, total sleep time estimates. It turns out that in wave one was conducted maybe 20, 30 years ago. The devices were very early versions, right, of these home-based sleep recorders. And so battery life, right? So often the studies would truncate. And of course, they're more likely to truncate people who have longer sleep, which is why these individuals, the slope is sharper here, right, than here. And so, we identified that as a clear problem. We also put a solution out, which is an analysis where you uniformly truncate all studies, some fixed number of minutes from sleep onset, gives you then at least conditional but apples to apples kinds of comparisons if you want to compare across waves and so on. So at least for the first, n hours of the night, and that resolves the apparent paradoxical associations, between and within waves. So just as an example of how, even the simplest measures can be impacted by study design. This is a similar one, I guess, in some ways. Okay, so the impact of the protocol on sleep architecture. So we just getting interested in this question of what were the protocols. Some tools, we developed a tool, Hypnoscope, just to visualize hundreds or thousands of hypnograms and to align them either by sleep onset or clock time and so on. And so you can kind of play around with these. But this just shows two studies where, let's see, so yeah, gray is wake. This is where the recordings start. So you can see something very clearly in this study. Pretty much everyone finishes at 6 a.m. In other words, the study protocol finished things at 6 a.m. Cleveland Family Study, likewise, a similar kind of truncation of sleep. We also see here, there's a kind of a curious little waking period here where people who went to bed earlier, right, they all magically awoke here around about 10 o'clock and then went back to sleep. You don't see that effect here. It turns out these just reflect study protocol. These were inpatient studies, and people they had a blood draw, right? So they were woken up here. So that's why that's there. And clearly the recordings were just stopped at this point. The primary goals of these studies was to say something about sleep disordered breathing. And in as much as that's the case, these factors don't really matter, right? It's not a badly designed study. It was a well-designed study to look at sleep disordered breathing in these populations. It's just to underscore the point that if you, though, want to take these data sets and use them to some other purpose, say to study something about sleep onset dynamics or how sleep duration and chronotype are associated, you can imagine these kinds of study protocol effects are going to have quite profound kind of impact here on some of the conclusions you may draw. So just as always, a kind of a caveat emptor for using some of these. Okay, so that aside, and I should say, despite these issues, there have been, dozens, hundreds of papers that have used NSRR data to successful and kind of replicated studies. And so I don't want to paint a overly negative picture. I just want to kind of point out some of the issues that one may face. So what else can we do with some of these data? So looking back, when we think about sources of individual differences in sleep, clearly age is one of the major sources here. The NSRR contains individuals from, I think, six months of age to like 96 years of age and so on. And we previously published, it's actually my first foray into the sleep world, was just looking across this age range and looking at spindle activity. And so we already knew that this changes, and other people had shown this, spindle activity changes in really quite profound ways over the whole life course. And so that's something we wanted to look at, of course, in the schizophrenia sample. And indeed, if we asked, how does, say, fast spindle density, how is that related with age? we actually found that in controls, it wasn't associated. And that's because of the age range here, right? These are between, say, 20 and 45 years of age. If we go back to this slide, the spindle activity really shows a kind of an inverted U kind of shape, right? Of ramping up during childhood, plateauing in young adulthood, and then during senescence and so on, in line with neurodegeneration, just declining here in older ages. And so we're really at the top of this plateau, right? It's not about to start going down yet. And so we don't see anything in controls, and that's largely what we'd expect. What we do see in patients, though, is a significant and really kind of moderate decline, even though they're matched for age here, and this is significantly associated. And so that was interesting, given some of the other work that suggests that the brains of patients with schizophrenia may show an accelerated aging from MRI studies and others, and so potentially we're seeing similar effects here. And so that's one way we can look at it, but really, what I'd like to do is try and use NSRR and other reference data to kind of basically have a tighter kind of coupling of previous data and model. What I mean, by example, the usual kind of data sharing model is you do some study and you share your data somewhere. And there are all of these things we can do to publish more papers with those original data. What I mean here is ideally, right, we would like to take those original data and learn from them in a way that we can interpret our second study in a better way, right? And so this is what I mean by reference-based models. And so in genetics, it's just everything you do is really informed with this kind of lens. If you collect data now, you would analyze it in a fundamentally different way, given all of the resources that exist about population genetics, the reference genome, and so on, than you would have if you collected the same data, say, 20 years ago. Versus with the sleep and sleep EEG, that's not really the case, right? Analyses are much more self-contained, and we don't try and leverage those existing data. Automated staging, I think, is one example of that, where you're trying to take existing data and directly use it to inform the analysis of your new studies. But there are other, I think, forms of that same kind of reference-based, model-driven predictive work that you can think of, as well as where artificial automated staging is kind of a special case of that. And so one in the sleep, but there has been others have applied in the sleep EEG, and we've seen in other areas of imaging and epigenetics and so on, is to predict so-called brain age from the sleep EEG. And so some colleagues in Boston did this successfully in adults some time ago, and we've implemented this. We did some work looking at this in the context of pediatric samples with kind of similar performance here. You can predict very accurately, with a correlation, say 0.93, somebody's chronological age as a function of the sleep EEG. But of course, what's important here then is the predicted age difference, right? This delta, which is a measure of potential delay or acceleration in brain aging, interpreted this way. And we showed that tracks with diagnostic categories here, say Down syndrome and intellectual disability. So applying this to the schizophrenia case, we indeed found that you could predict somebody's adult age in both cases if we use this previous model trained on these features. And so, you get reasonable performance. On average in controls, there's no bias, right? The difference on average is 0. Versus in patients, you see the same kind of slope, but we are, the predicted age is on average about 5 or so years older than expected. And as I said, that kind of connects to some of the literature from other brain imaging modalities and so on. Now,
Sean, can I ask a question?
Yeah, sure.
What can you even give us a brief idea of what were those features in the non-REM EEG that predicted a more aged brain?
Yeah. So basically here you can do this various ways, but here there are 13 features, and really it's pulling out the features of the sleep EEG that independently track with age in the strongest way. And so, delta power, spindle density, a measure of coupling between slow oscillations and spindles, some of them slightly more esoteric measures of so-called spectral kurtosis. It's really kind of black box, right? It's just taking whatever set of features and just using it as a way to summarize those measures and come up with one number that I think in some cases can be useful. You probably could go and pull out a different 13 features and have a model that performs, similarly. So, it's an interesting question, right, of how much you interpret any one feature here. But if you're seeing these effects, typically it's not driven by like one element of this, say spindle density. When we're seeing this consistent differences, there's a kind of a coordinated set of changes of all of these things that show marked age-related declines in the typical population. All of them to some extent are showing that same effect in patients. So we'll get back to that a little bit, right, when we think about what drives it. But so age is one thing we expect is going to have a profound influence on somebody's sleep architecture and their microarchitecture. Something else that obviously in the context of a psychiatric disease, schizophrenia, is going to be a profound problem, right? Is medication effects as well. All of these patients were taking an antipsychotic at the least, and maybe multiple. And indeed, we found that the details here aren't important, but these are some of the measures of sleep macroarchitecture and some spindle measures of coupling, slow oscillation metrics, some other power and connectivity measures summarized here. And what this is showing is associations after adjusting for multiple testing with the different antipsychotics and other classes of medication. The point here is that, and these are the group differences with respect to controls at the bottom. If you just zoom out, the bottom line is among patients, there's a lot going on here, and it's kind of complex in terms of some of these medications pushing something up, some of them pushing something down. Say, for example, olanzapine and clozapine have statistically significant but opposite direction effects on N2 duration. If you remember that first slide where I showed you there was greater variance among patients for N2 duration, obviously, is that just trivially reflecting the different medications one is taking? Even though these all nominally have similar targets, right, of just kind of D2, obviously they have different off-target effects and so on. And so they seem to be generating differences among patients that we're picking up here in the sleep EEG. And so is that, what's going on there? Can we control for that? And so, it's tough to control for, but we try to, and at least taking advantage of the fact that all patients are taking something, but different patients are taking different types of medication. This was the result we had originally, right? This is where we ask, how many of these metrics out of whatever we tested, let's say 4,500 metrics, how many of those showed increased variance among patients? And the original answer was about a quarter of them showed more patient-to-patient variability, right, than control-to-control variability. If we tried to adjust for the effect of medication before asking that question, we find it goes down here. Okay, yeah, and in contrast, hardly any of the controls had increased variability. If we adjust for common medications, this goes down about half, but this is still much, much more than you'd expect and much, much more than the controls. Okay, and so it's some of this effect here is driven by medication, but not all. I should say, one of the first things we looked at, does this variability relate to clinical and cognitive symptoms? And the answer was no, not at all, right? And so, it's telling you something different. Potentially some of this is something about just medication. It's sensitive to medication in a way that those other measures aren't. But potentially, given we know these are heritable measures, some of this is still reflecting potential etiologic differences between patients. But we wanted to explore the medication part, and we took this back with regard to the accelerated aging we had seen. So this was that original result. I showed you the graph, but just summarizing it as a table, right? On average, the patients were something like 5, 6 years older than you'd expect, right? Given their sleep EEG. That wasn't the case in controls. But just splitting out the patients here by, this was the most commonly endorsed antipsychotic here, olanzapine. And here we found, the patients then, there was a big difference just between those two groups that was statistically different here and a similar order of magnitude than the case control difference, right? And so there's another 5 years for just being on olanzapine. This didn't seem obviously related to duration of illness. We controlled for age, we controlled for other clinical factors, okay? And so, it's kind of, in a sense, concerning, right? If you think of it as a confound in these kinds of studies, there's such large effects on the sleep EEG. But I think there's another way to interpret these effects that connects to another literature, which is we know that taking antipsychotics is key for patients, right? To reduce, positive symptoms. But there's a known just negative side effects on cognition. And some studies have shown that, sustained use of antipsychotics and other forms of drug over time can lead to, fundamental changes in the brain, cortical thinning, things that are related to aging, and especially that this effect can be exacerbated in older patients. And indeed, if we stratify our study by older or younger here, we see stronger effects of olanzapine on brain age in the older individuals than we see in the younger individuals. And why that becomes important is, to what extent is the sleep EEG reflecting, a measure of sensitivity to the deleterious, potentially iatrogenic effect of medications on cognition? To what extent can we use that as a biomarker to predict individuals who may be more sensitive to the negative side effects of certain medications? And also, to what extent can we, in mice, then use these in a way that we want to design new therapeutic agents that don't have this impact, right, on brain aging? And so this is something that could be, in principle, transferable to animal models, right? If your goal is to find better antipsychotics that don't have cognitive side effects, right? This, I think, could be, here, the sleep EEG could kind of be a powerful bridge. We took this a little further. I'm mindful of, I'm getting low on time, so I'll try and wrap it quickly. We took this a little further just out of the context of schizophrenia to ask, do we see this effect more generally? And so something we noted in the schizophrenia case, the medications that had the stronger impact on brain aging had typically higher anticholinergic burden. And such that, there's very standard ways to kind of characterize an individual's anticholinergic burden. We used this ACB measure. Just over half the sample have so-called high ACBs, and we'd expect that, because there's a lot of polypharmacy and all these people are on strong medications. Of course, there are other populations that can have relatively high rates of anticholinergic burden too, due to polypharmacy, and elderly populations is one of these. So we looked at a study, Mr. Ross. It's about 3,000 individuals in their kind of 70s and 80s, and indeed found about 10% of these individuals had so-called high ACB, kind of rated this way. And, this was one of the studies I alluded to earlier in the study of cognition and the sleep measures. And if we go back and look at this, indeed, we find in Mr. Ross, having just a higher ACB predicts both older brain age from a sleep EEG, but also worse cognitive performance on the measures we looked at, and also tracks with some of the measures, not all of them, but some of the measures we previously kind of flagged, right, in the Mr. Ross study as being associated with poorer cognition here, REM sleep, spindle density, and so on. Obviously, there's confounding by indication, and so it's challenging to kind of pull these things apart completely. But for what it's worth, if we try and control for age, sex, race, and then BMI in common diseases, these effects were kind of broadly seen. And certainly it's consistent with studies looking at the acute effects, right, of various medications administered can have quite profound, but also quite diverse effects on sleep microarchitecture. And so the extent to which we're picking those up, right, versus picking up the iatrogenic side effects that are downstream from prolonged medication use, to what extent that is driving, right, some of these relationships we see with sleep and cognition, I think is one form of interesting question. But I said as well, you can also think of this in terms of, can we use this information to, say, predict individuals who may be at higher risk of cognitive symptoms and so on from medication? So I think there's, that from my perspective, a lot to be done in this area, but there's definitely a kind of a complex set of associations here. I'm going to whiz through these just because I know we're kind of out of time, right? So I apologize, but I'm going to zip through and just make the basic point, rather than just looking at whole night averages, right? There's a whole structure of just dynamic processes. I show examples where we can look at ultradian rhythms at the cycle level, so-called infraslow oscillations as well. These are areas where there's a lot of emerging kind of basic science and mouse work. And these are things that are detectable, kind of sitting a time scale atop, if you like, of what we're looking at typically when we look at slow oscillations and spindles. To cut to the chase, we see some pretty profound but independent effects in disease groups and so on with regard to these dynamic measures, as well as these whole night averages. And the important point is, we're doing this not just to that original AUC for the schizophrenia prediction, right? It was already kind of maxed out of 0.94, 0.95. The goal isn't just to try and build a incrementally better classifier of disease or even brain age and so on. The goal here, and I think this will really be the challenge, is to try and disentangle, what may be an effect that's driven by disease state versus what may be driven by natural aging versus what may be driven by a medication effect. And so the goal here is any one measure may have a similar profile of effects with respect to those factors. But the goal here would be to find kind of disambiguating distinct profiles where you see something different, say in infraslow oscillation space, than you do see looking at things classically. And there's some suggestion that, indeed that's what we see. I'm going to skip through these slides. This is medication effects on, for example, infraslow rhythms. And so obviously I can't talk through that, but these 2 points here was summarizing what I was trying to say with that. So I apologize for going over and not getting a chance to speak to that. So, the bottom line, it was taking this one entry point into looking at sleep biomarkers from this kind of, almost now classical literature of just spindle deficits and schizophrenia. We expanded out in a larger sample. We found a more kind of complex set of variations that are reliably associated at the group level. But then we find a lot of variation among patients. And to some extent, that reflects aging and accelerated aging, and medication effects and other factors. But of course, the goal here would still be, to what extent is it going to tell you something that's, ideally clinically actionable, something that is reflecting genetic differences, and something that you can have a kind of a translational bridge to some of the animal work that my colleagues are doing in mice, looking at these same kind of outcomes. The goal here, ultimately, that these multi-scale analyses will provide one way of disambiguating these kinds of effects. And so, yeah, I apologize for kind of whizzing through that last part, but I just at least wanted to try and give a flavor of some of that biomarker work, and at least some of the ways in that some of the population, the NSRR and other types of data are kind of we use to inform some of that. These are just some of the tools that are available, and these are some of the my colleagues, in particular, Natalia Koziemiakou and Jen Pan for the GRIN study, and the founder of the NSRR, and my colleague on much of this, Dr. Susan Redline. So I'll stop there. Thank you, and apologies again for going over.
Thank you. That was fantastic. I think you're getting some kudos in the chat as well. I don't see any additional questions in the chat. I don't know if anyone wants to unmute with more questions. I know people were able to ask questions during the session. I don't see any additional questions, but thank you again. That was fantastic.
Excellent. No problem. Thank you.
Take
It's a
care.
pleasure. Okay. Bye-bye.
Bye-bye.